Hello students. Today I am going to give a presentation on history of a biotechnology. Students, you might be knowing that definition of a biotechnology, that is, biotechnology means any technological applications that uses biological systems, living organisms, or derivatives thereof to make or modify the products or processes for specific use. This is one of the most important, we have to say that, the definition of the biotechnology, or the controlled use of a biological agents such as microorganisms or cellular components for beneficial use. Friends, actually this definition gives us idea related with very interesting thing is that here we are going to use the microbes, or we are going to use the living organisms, isn't it? And it's, we have to say that, secretions to modify, we have to say that, the products or sometimes the process, in the sense we are going to make that very good energetic, very nutritious food, very, we have to say that, that is actually modification. And in processes, just to increase the speed of process. For example, we know that most of the enzymes are present in the microbes that we can use for the fast processing of a, we have to say that, systems or any products. So biotechnology is, that is nothing but, a bio means living, related with that one is there, and technology means technique, isn't it? So technological applications, isn't it, that uses biological systems, living organisms, or derivatives thereof to make or modify the products or processes for the specific use, in the sense, in the benefits of a human being. Students, today I am going to give you idea about history of biotechnology. Now you might be little bit in the confusion why to study the history. We are not the history student, as we know that in case of the art faculty there is, to the art student there is one of the main subject, or I have to say that in the FYSY there is a subject of a history is there. Then what is necessity to learn the history? Friends, keep in mind, आपल्याला माहिती आहे की इतिहास तेच लोक घडवतात, those who are, we have to say that, little bit mad, isn't it? Mad in the sense, not creating problem to the society. Those who are creating such type of things which could be the benefited, which could be the beneficial to the generation after generation, isn't it? म्हणजे येणाऱ्या पिढ्याने पिढ्या या लोकांना तो इतिहास सांगत असतो की आमच्यासारखं बनत चला. सिमिलर वे, जरी आपण शिकत असलो प्लांट बायोटेक्नॉलॉजी, तर बायोटेक्नॉलॉजीचा सुद्धा एक इतिहास आहे. तो इतिहास घडवणारी माणसं होती, मी सायंटिस्ट वर दॅर, अँड दिस सायंटिस्ट हॅव टेकन अ लॉट ऑफ इफर्ट्स just to reach up to such a type of achievements, and we are enjoying all these things right now. They have made the history, and after reading history, after going through the history, we can also make the history. म्हणून इतिहास दोन गोष्टीसाठी वाचायचा. एक, ज्यांनी इतिहास घडवला त्या लोकांना स्मरण करण्यासाठी, जस्ट टू रिमेंबर दोज पीपल्स, दोज हू हॅव गिवन ईच अँड एव्हरीथिंग टू द सोसायटी of their life, isn't it? Those who have given each and everything of their life to the society. They have given total their life. Only they live for the society or to the human being. One. Another thing is that after going through this history, that can inspire to us, that history can inspire to us, to you, to me, just to think about the plant biotechnology and make the history. then people can remember to us in future. So, with these two things, you might have come in your mind that why to learn the history of each and everything. So today we are going to learn the history of a plant biotechnology. Isn't it? We are going to learn history of plant biotechnology. Here, my friends, you might be coming that in 19,002, Haberland proposed the concept of in vitro cell culture. Means what? In vitro means in a test tube. In test tube actually, he started growing up a cell with the help of a culture medium. So he proposed in 19,002. Again in 19,004, Hanning cultured embryos from the several cruciferous species. Cruciferi is the family. We know that मोहरी जी आमटी करत असताना छोटे छोटे ग्रॅन्युल्स आपण त्याच्यामध्ये टाकतो. फेंट ब्लॅक कलर ती मोहरी आहे ती त्या फॅमिलीची आहे. त्या मोहरीतले एम्ब्रिओ काढले. मोहरीच्या फॅमिलीमध्ये असणाऱ्या प्लांटचे एम्ब्रिओ काढले. कुणी? हॅनिंगला इन 19,004 and cultured that embryos. Again in 19,022, Kolte and Robbins successfully cultured root and stem tips respectively. Friends, you have to keep in mind, we know that there are the three main types of the meristems. Isn't it? Apical meristem, that is well known to you. Why? That meristems are nothing but the cells having capacity to undergo the division and redivision. So, Kolte and Robbins successfully cultured the root and stem tips respectively because the root tips and stem tips shows presence of a meristems. And that meristems, they have selected properly and cultured in a test tube with the help of a culture media in 19,022. In 19,026, Wendt is the name of scientist, discovered first plant grown hormone, that is IAA, indole acetic acid. Keep in mind, my friends. In 19,026, Wendt discovered the first plant growth hormone. So, in culture medium, before that one, that was not used in the culture medium to grow the plant or to use for the growth of a cell. But when Wendt discovered that in 19,026, IAA started to use in a culture medium at the time of growing of a cell and plant in vitro. Isn't it? Now, in 19,034, White introduced the vitamin B as a growth supplement in the tissue culture medium for the tomato root tip. Now, what happens? You see, students, this cannot be done within a 1 day, 2 day, or 3 day. Lot of reading, lot of efforts, a lot of experimentation have been carried out by these scientists just to reach a single sentence, just to tell very simple conclusion that vitamin B can be used as a growth supplement in tissue culture. Lot of or many days have been taken. Now in 1939, Gowhart, White, and Nobokot established endless proliferation of a callus culture. As we know that callus is nothing but unorganized, undifferentiated mass of tissue. So, these three scientists, Gowthart, White, and Nobecoat established endless proliferation of a callus culture means they have grown the callus on a large scale. In 1941, Orbach was first to add the coconut milk for the cell division in the Datura. Keep in mind, friends, natural things. Just to think on that one. Although right now we are giving a coconut water to the patients, we are providing because that is a shows presence of a energy, isn't it? After having that quick energy can be made available to the person, those fellow who have had that coconut milk, we have to say that. So, Orbeck added coconut milk for the cell division in the Dhatura because that coconut milk shows presence of a growth hormones or supplements or vitamins which are required for the growth of a plant in vitro on culture media. Now, Ball raised the whole plant of the lupinus by shoot tip culture in 19,046. You see actually this journey of a growing of a plants, a growing of a cell in vitro started in 19,002, means in the beginning of a 20th century. But in half of the 20th century, whole plant of lupine is grown with the help of a shoot tip in vitro. In 19,054, Muir was first to back a callus tissue into a single cells. Isn't it? That callus is a mass of unorganized cells, but just to separate the cell will help to proliferate, just to multiply, just to develop many plants with the help of a small amount of a callus. So that is actually started in 19,054 by the efforts carried out by Muri, breaking of a callus. Now Skoog and Miller discovered the kinetin as a cell division hormone in 19,055. Skoog and Miller, they have discovered the kinetin, which is a very much useful for the division of cells, and that kinetin hormone is discovered by the Skoog and Miller. Again in 19,057, the Skoog and Miller gave a concept of hormonal control of the organ formation. In the sense, friends, you have to keep in mind, from the callus, first of all, the shoot gets developed. That is depend upon the proportion of a auxin and cytokine, isn't it? Suppose we want to make that callus to develop the root system, also we have to change the proportion of a auxin and cytokinin in the growth or culture medium. So organ formation, Skoog have given that concept in 1957. 1959, Reinert and Stewart regenerated embryos from the callus clumps. Keep in mind, cell has been kept or shoot tip has been kept on the culture medium in a test tube, and later on that is grown into a callus, and again from the callus embryogenesis have been done. When organ develops from that callus, that is known as organogenesis. But whenever embryo develops from the callus directly, then we have to say embryogenesis process. In 1959, Reinert and Stewart, these two scientists have developed. In 1960, Cocking was the first to isolate the protoplasm by the enzyme and degradation of the cell wall. Keep in mind, my friends, in 1960, very interesting, we have to say that methodology developed by the Cocking for the isolation of a protoplast, and very, I have to say that advantage of this method of isolation of a protoplast by using enzyme is that without injured. 100% guarantee of getting a more and more protoplast, healthy protoplast with the help of this method. So isolation of a protoplast method is very useful method given by the cocking scientist to us. Now, Guha and Maheshwari in 19,066 produced first haploid plants from a pollen grains of a dhatura. We know that actually. Reproductive parts, that is pollen grain, is a haploid one. Whenever pollen grain fuses with, or we have to say male gametophyte fuses with female gametophyte, it becomes a diploid after fertilization. They develop into a zygote, diploid zygote. But directly from the pollen grain, they have developed the haploid plant. There is no diploid plant. Keep in mind, number of chromosomes is a, we have to say that the half, not the double, single. Now in 1970, Smith and Nathans discovered the first restriction enzymes from the Haemophilus influenza. You see how the biotechnology is going to get developed from 19,002 to 19,070. Restriction enzymes. These enzymes are used to break the DNA at a particular restriction site, isn't it? So that is nothing but we have to say that very interesting history is that to this biotechnology. In 1970, actually, one of the Haemophilus influenza restriction enzyme is discovered by the Smith and Nathan. In 1970, Baltimore isolated reverse transcriptase from the RNA tumor viruses. These things are very useful for the diagnosis of diseases, very useful for the development of a antiviral. I have to say that the vaccines. Baltimore isolated reverse transcriptase from the RNA tumor virus in the sense from mRNA we know that is developed. Messenger RNA developed on the RNA. But from the RNA, from the messenger RNA, again RNA is to be developed. In 1972, Carrison produced first interspecific hybrid of a Nicotiana by protoplast pigeon. Friends, you see, isolation has been done in the 1960, but it takes actually 12 years just to fuse that isolated protoplast for the production of a hybrid variety. Genes of one variety mix with genes of another variety and then develops a third generation. That is the actually from these two combinations we develop the new, we have to say that the variety that is hybrid variety. 1972 again Berg produced a first recombinant DNA combining SV40 virus and a virus. Now in 1974, Janet and its co-workers discovered tumor-inducing plasmid, which is used, that tumor-inducing plasmid. We know that plasmid is nothing but the actually double-stranded, self-replicating, circular DNA which is present in the actually bacteria, and that actually isolated from the agrobacterium, that is actually isolated from the bacteria and induced into a agrobacterium. The agrobacterium have capacity to insert the recombinant DNA into the monocot plants or the plants which are not having root nodules. So with the help of this one we can make plant a root nodule having, with the help of this technology. We know that root nodule shows presence of a nitrogen fixing gene into that one, or rhizobium bacteria which fixes atmospheric nitrogen. But such a type of things present in the nature, but artificially we can make monocot plant, those which are not having actually originally the root nodules on their roots. So this is also done because of the discovery of a Chenin and his co-workers, not Chenin et al. 1977, Chilton and his co-workers successfully integrated TI plasmid DNA from Agrobacterium tumefaciens into the plant. Actually, no development has been done, but in 1977, scientists got the success because of the efforts taken by the Kilton and et al. And his co-workers, I have to say that we succeed. Scientists got success to introduce a tumor-inducing plasmid into the roots of a plant with the help of Agrobacterium tumefaciens. In 1977, Sanger, Maxim, Gilbert gave a technologies for the DNA sequencing, very interesting, in 1977. So friends, you might have now understand how the history has been made by the biotechnologists and why to remember them, because they were mad behind all this type of research, and that research is going to help to the human society over increasing population, again to solve the many pollution problems, isn't it? These things are going to help to us to increase the yield of plant. But for that one, lot many, we have to say that the many scientists have taken their efforts, and that's why we have to learn the history. We have to remember the persons, the fellows, the scientists, the we have to say the honorable, all these fellows. And again, that things inspire definitely us to make the history by doing the research, by working for the society, by serving to the society with the help of our research. Now, Carey Mullis invented polymerase chain reaction (PCR) for the amplification of DNA. We know that we require always not a single DNA strand, but we require that in a huge amount, in a large scale at that time. Polymerase chain reaction helps to us. So that is given by the Carey Mullis in 1983. At the end of, we have to say that, the 20th century. In 1984, Horsett and his co-workers developed a horse, not a horseshead, horse developed the transgenic tobacco by transformation with agrobacterium. In 1987, Clean and his co developed a biologistic gene transfer method for the plant transfer. Biologistic method in this gun method, that is also shotgun method is that. With the help of a mechanical instrument, the gene can be introduced into the nucleus of a cell, and after growing that cell into a whole plant, that becomes a hybrid plant. So shotgun method physical. So such a type of a history is there. Again, in 1960, Bergman filtered cell suspension isolated single cell by plating. In 1962, Murashige and Cook developed a MS medium with higher salt concentration. Murashige's Cook medium, culture medium is there. In 1962, Kanta and Maheshwar developed the test tube fertilization technique that is used right now, my friends. Whenever there some, because of some what I have to say, the problems unavoidable problems, the couple cannot give a birth to their next generation. At that time, this technology is useful. In 1966, Stewart demonstrated totipotency by regeneration in carrot plant from the single cell of the tomato. This Stewart actually have found one of the most important characteristic of each and every cell, that is a totipotency. Totipotency is nothing but the potential of the cell to undergo the division and redivision and to develop into a whole plant. That is a totipotency. Now in 1995, Fleischmann et al. Sequenced the Haemophilus influenza. Again, in 1997, Blatner sequenced E. coli genome and in 2001 Human Genome Project Consortium and Venture and his co-workers sequenced the human genome successfully. In 2005, Rice Genome sequenced under the International Rice Genome Sequencing Project. So these things will be help you to understand gene sequencing. Actually, suppose one example I will give to you which is related with our own is nothing but the suppose the 10-year guy after 40 years, after 50 years, which type of disease may can affect to this fellow that we can understand at the age of his 10. So before that one, some preventive measures can be carried out for 30 years before the 40th years. And that disease can be, we have to say that eradicate or to overcome the problem of that disease. So that is possible because of sequencing of a gene. Many more things are there. So friends, you might have come to know that. Why to learn the history? दोस्तों मैं हमेशा कहना चाहता हूँ जो पीजे कलाम हमारे राष्ट्रपिता राष्ट्रपिता नहीं ये इनको हम कहेंगे प्रेसिडेंट फॉर्मर प्रेसिडेंट ऑफ आवर इंडिया राष्ट्रपति उन्होंने बहुत अच्छी चीज़ कही है realinspiration4you.com पे अगर सूरज की तरह चमकना चाहते हो तो सबसे पहले सूरज की तरह तपना सीखो दोस्तों माय फ्रेंड्स द साइंटिस्ट हैव टेकन अ लॉट ऑफ एफर्ट्स वो तप गए थे और इसके कारण वो चमक रहे हैं इन द सेंस वी आर रिमेंबरिंग टू दैम बिकॉज दे हैव टेकन अ लॉट ऑफ एफर्ट्स दे हैव नॉट थॉट अबाउट दैट सोसाइटी व्हाट मे कैन गिव दैम बट दे हैव गिवन लॉट ऑफ थिंग्स टू द सोसाइटी एंड इसलिए एपीजे कलाम ने भी ये कहा सूरज की तरह चमकना चाहते हो तो सबसे पहले सूरज की तरह तपना सीखो ही बायोटेक्नॉलॉजीची हिस्ट्री वाचून आपण त्यांना आठवलं स्मरण केलं या शास्त्रज्ञानं त्यांच्या कामाला त्यांच्या नावाला बऱ्यापैकी आपण ॲप्रिसिएट केलं माझी इच्छा आहे भविष्यात हाच इतिहास वाचून तुम्ही इतिहास घडवाल ही मला अपेक्षा आहे आणि या अपेक्षा आपण पूर्ण करा याची मला खात्री आहे थँक यू व्हेरी मच विथ दिस नोट